Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Martin Kriegel. Dr. Kriegel is Chief of Rheumatology and Clinical Immunology at the University Hospital of Münster in Germany, and Associate Professor Adjunct of Immunobiology at the Yale School of Medicine. Dr. Kriegel's research showed that while Lactobacillus reuteri can drive autoimmunity, it is ameliorated by dietary-resistant starch. Today, Dr. Kriegel presents his session, Dietary-Resistant Starch Effects on Gut Pathobiont Translocation and Systemic Autoimmunity. Dr. Kriegel, over to you.
Hello, everyone. I'm Martin Kriegel, and I'd like to talk about dietary-resistant starch effects on gut pathobiont translocation in systemic autoimmunity. These are my faculty disclosures. I would like to start with a brief introduction what my lab has been studying over the years. And a major question we'd like to answer is, how does autoimmunity arise? Which is, of course, a very broad question because we have the genetic predisposition that contributes to autoimmune disease. We have at the core of these diseases immune dysregulation of the innate and adaptive immune system. But there's always this ill-defined environmental component that we all know about. Over the last few years, it became clear that it also involves our microbiota that are influenced by the environment. And we focus on the gut and skin microbiota that we are colonized with. And specifically, we focused on non-gut autoimmune diseases such as lupus. It's a systemic autoimmune disease that is affecting many organs except the gut. And we wanted to understand how the microbes in the gut can interact with the immune system outside the gut to potentially contribute to these diseases. And what we've uncovered over the last couple of years is that there are several cellular and molecular mechanisms. In these first two papers that I mostly will talk about today, we focused on the possibility that bacteria can just leave the gut and breach the barrier and get into lymphoid tissues and then interact directly with the immune system. And this, of course, also happens on the basis of a genetic predisposition. And in this autoimmune disease called lupus, genes related to the toll-like receptor 7 pathway and related genes is important. And in a mouse model that I will talk about with overexpression of this TLR7, we could see that the barrier was leaky and microbes could translocate and instigate autoimmunity. Another mechanism on the molecular level that I will not talk about today is cross-reactivity with antigens in the microbiome, so that the immune system gets confused and attacks not only the microbe, but also self-antigens in the body. And these two papers I just list here for anyone who's interested dealt with this important paradigm that cross-reactivity could be a chronic trigger in certain autoimmune diseases. Now, for the sake of this talk, it's important that we also test certain interventions in animal models and plan to do this in humans too. And one important one is, of course, diet. Every dietary component essentially will influence the gut microbiota. And I will tell you in the next 40 minutes or so a story how we dissected host microbiota interactions in the setting of a dietary intervention. First, as an introduction, on lupus, systemic lupus erythematosus, or SLE, we take as a paradigm to study host microbiota interactions. It's also an interesting clinical disease that affects many different organs, such as the kidney, the lung, the skin, the brain, but rarely, if ever, the gut. So it's also a conundrum how perhaps gut microbes could contribute to this disease that mainly happens outside the gut. And that brings me right to the one paradigm I alluded to in the introductory slide, the leaky gut, which is not a great term. We should say gut barrier dysfunction. But this has been in the lay press for many years, but only over the last 10 years or so, also rigorous science has been conducted and clearly showed that there is an important contribution to a variety of chronic diseases, including autoimmune diseases, if the gut is leaky. And for a long time, it's known that molecules, toxins can get through the gut barrier, get into the blood and lymphatic vessels and spread. But what we really focused on is if whole bacteria, viable live bacteria, not just 16S signals or particles, can get through the gut barrier and instead of an infectious disease can cause autoimmune disease. And as mentioned in these two studies that I'll touch on, we identified certain lactobacilli and enterococci that specifically can translocate and drive autoimmune pathways. And it's important to note that we isolated specific strains, while the field as a whole has also shown that microbiota can translocate in inflammatory settings. But to us, the key question is always which individual bugs is it and how do they interact with the immune system? And this has not only been studied in the autoimmune field, also in cancer. There have been around the same time two very important studies by others clearly showing that gut microbiota translocation contributes to lymphomas, as Meisel et al. Has shown here in Nature a couple of years back. And this other study a year later showed that, interestingly, not just the gut bacterial microbiota, but the fungal microbiome contributes to pancreatic cancer by translocating from the gut to the pancreas. I just thought I'd mention these interesting studies, as this is an emerging and rapidly evolving field of the gut microbiota translocation. And in fact, it's, I would say, even more heavily studied in the cancer field. And of course, again, the specifics matter, which bugs translocate and which cancer one studies. And these three papers I cite here nicely show that translocation of bacteria can also be beneficial in the setting of cancer, providing anti-cancer immunity. And in fact, one bug we isolated here, an Enterococcus gallinarum strain, I'll briefly talk about in a minute, is currently being developed by a biotech company in Ireland to use as anti-cancer immunity in patients with cancer. So with that, I hope I've given you a brief overview of this paradigm, because we studied this then in the context of a dietary intervention, which becomes important later. So we started out to study this phenomenon first in a certain lupus-prone animal model where we saw there's dysbiosis, and importantly, there's an impaired gut barrier compared to a healthy host with a normal mucus layer and gut barrier. And we saw that this gut pathobiont called Enterococcus gallinarum is a major bacterium that translocates to the secondary lymphoid organs as well as to the liver, the first firewall after the gut, through the hepatic portal circulation. Particles, or even in this case bacteria, can reach the liver. It can induce their autoantigens and innate immune pathways. And in lymphoid organs, it also can skew the immune system to pro-inflammatory responses such as T helper 17 cells, and they can stimulate T follicular helper cells that we've shown can then aid in immune complex formation by autoantibody production against these antigens. Lastly, there's an important innate immune pathway in lupus, in this systemic autoimmune disease called type 1 interferon from alpha and beta, that's primarily produced by plasmacytoid dendritic cells, or PDCs. And for the rest of this talk, I'll focus on this PDC axis. Sorry, I just wanted to also mention that besides monocolonization studies with this specific bug, we also, of course, successfully prevented lupus with an oral antibiotic that depletes this Enterococcus. And importantly, we also developed an intramuscular vaccine that specifically targeted Enterococcus gallinarum and could also prevent lupus from developing. But what patients always ask is, of course, is there a diet that I can already use to improve my microbiome? And this is, of course, a very difficult question, but we tried to get first answers by dissecting another mouse model for lupus. And now I get to this important PDC interferon axis I mentioned earlier, because while we were dissecting this translocation paradigm, we studied a TLR7 transgenic mouse model for lupus with a diet intervention. We obtained these mice that overexpressed TLR7, toll-like receptor 7 by Sylvia Boland from the NIH. And this is a great mouse model that she developed because it's at the core of also the pathophysiology in patients with lupus, where plasmacytoid dendritic cells are activated through these toll-like receptors like TLR7. They spew out type 1 interferon that can also further stimulate B cells that produce the autoantibodies, often against apoptotic particles and nuclear antigens. And then there's this vicious cycle that already is induced by this genetic overexpression in the mouse model and by the heterogeneous polymorphisms that patients carry. But we asked the question if through the microbiome we can, by applying a certain dietary intervention, we can break this vicious cycle, specifically by ameliorating this pro-inflammatory PDC type 1 interferon pathway. And first, I'd like to give a brief overview of diet, shaping not just the host, but also the microbiota, thereby shaping the host, which is, of course, well studied by many in the field. And I just give a brief evolutionary perspective here. Millions of years ago, we, of course, had mainly a vegetarian diet with increased bacteroidetes. Then we became hunter and gatherer and started a carnivorous diet with an increase in firmicutes. Over several 100,000 years, probably the best diet, the healthiest diet was the mixed constant diet with an increased microbiota diversity. And as many of you know, in modern chronic illnesses these days in industrialized countries, we see a loss of this microbiota diversity, which is detrimental in many ways. And part of the problem is this so-called Western diet, high fat, high sugar. It is low in fiber. And of course, it's well established by a great work from many people in the microbiome field that this can contribute to metabolic diseases and obesity. Now, we asked if a certain type of fiber called resistant starch could also be beneficial in inflammatory diseases. And we specifically focused on a so-called high amylose maize starch, which is a special resistant starch type 2 that we can give to mice in a controlled setting. And this resistant starch that we fed these TLR7 transgenic mice, it's called resistant because it has a different structure than the regular, more unhealthy starch that is cleaved in the upper gastrointestinal tract and then leads to glucose increase. But the resistant starch is fermented. It gets through. It's resistant to digestion in the small intestine. It gets into the large intestine. It is fermented by good bacteria that then have an effect on the host. And since we focus on the microbiome, we felt this is an interesting dietary approach to study. And indeed, Daniel Segarra-Ruiz, the PhD student on this project, showed that resistant starch feeding in the TLR7 transgenic lupus mice led to profound improvement in mortality of this otherwise lethal, spontaneous lethal lupus mouse model. And he tried two different starch diets. I show here green bananas because the human correlate of this resistant starch type 2 would be green bananas or raw potatoes, so nothing that's easily eaten, but I'll get to that a little bit later. But in this experimental setting, we could feed a high amount of resistant starch and had all other dietary components equal in the control group and could improve lupus mortality significantly. Now, the key question is, of course, how and what are the mechanisms? The first question is always, does the dietary intervention alter the gut microbiota? I would argue every diet we eat will change your microbiome. And not surprisingly, I show you here just for a few mouse cages an example. Over 6 weeks, of course, the microbial communities diverged. And we've seen this many times in the microbiome of these mice. But the important part, or at least what our lab focuses on, is which specific taxa is it and do they matter? And that's what the story today is about. And here, sequencing was actually useful, while it's very complicated in human patients. I'll also get to a little bit later. Here we saw persistently that in the lupus-prone mice, shown here in red, the TLR7 transgenic, we had consistently lactobacilli grow out, specifically Lactobacillus reuteri, interestingly, which you may rather know as a probiotic, but this particular strain was growing out in these mice. But in the mice fed with the resistant starch, shown in green, several good bacteria, among them Clostridialis, was actually enriched. And this outgrowth is shown here specifically for this Lactobacillus species, where you see from day 0, week 0 of the intervention to week 6, there was an ongoing outgrowth in the wild type TLR7 transgenics compared to wild type like 6 mice that had no lactobacilli. And importantly, when we fed then these mice with resistant starch, in green, you can see there was significant suppression of the outgrowth of this bacterium. And at that point, we had no idea if this is just an association or it had something to do with the mechanisms that lead to lupus. But as mentioned earlier, around the same time, we uncovered this translocation paradigm and also tested this in these mice. And indeed, in the mesenteric lymph node, liver, and spleen of these TLR7 transgenic mice, we saw persistently translocating Lactobacillus reuteri into these secondary lymphoid organs. So enterococci and lactobacilli, we know from old studies on mice that were put under stress, they seem to be preferentially to translocate into tissues. And in the one lupus mouse model, we saw an enterococcus isolate. In this mouse model, this lactobacillus isolate draw. But most importantly, we could link this phenomenon to the pathogenesis in lupus. And so it was not just an epiphenomenon, this translocation. In lots of immunology data, which I just point you to this paper, Daniel could show that the translocation was driving the systemic PDC type 1 interferon axis with increased interferon alpha levels. So the already genetically heightened interferon levels because of the TLR7 overexpression was further aggravated by the microbiome, specifically by lactobacilli that translocated. Now, most excitingly, the resistant starch diet inhibited this process. So Lactobacillus reuteri translocation, shown here in red in the transgenic mice, in the lupus mice, compared to no translocation with sterile organs in the wild type, was significantly suppressed, shown here in green, with the resistant starch diet. And fitting with that, the so-called plasmacytoid dendritic cells that are enriched in the spleen and other systemic organs in the lupus mice were significantly and profoundly suppressed by the starch diet. So we think this is how the diet helps in the lupus model, but the exact mechanisms were still unclear. So you may well know that different fibers will increase your short-chain fatty acids by fermentation of these good bacteria, such as Clostridialis. But the conundrum was that Lactobacillus reuteri is in the small intestine typically, and the fermentation of the resistant starch should occur in the large intestine. So we hypothesized that perhaps there's diffusion of the short-chain fatty acids to the ileum, or that there's some produced also in the ileum. And so we first asked the question, does resistant starch increase short-chain fatty acids in these lupus-prone mice, as it's been shown in healthy hosts previously by others, that fiber in general should increase these acids like acetate, propionate, and butyrate. And indeed, I show you just one example, not just in the colon, but in the ileum. We saw a significant enrichment of short-chain fatty acids in the lupus-prone mice fed with resistant starch. Interestingly, the wild type fed with resistant starch as a control had no increase in the small intestine, just in the colon. There's something special about the microbiome and the geographic location of short-chain fatty acid productions. And fitting that these short-chain fatty acids are only produced through the microbiota, the germ-free mice with or without resistant starch had no short-chain fatty acid production. So we think that the Clostridialis and other important short-chain fatty acid producers are the ones that then produce butyrate and other short-chain fatty acids that then have beneficial effects on the host. And we saw that in the healthy mice, their Clostridialis are elevated and they're lost in the lupus-prone mice, while I showed you that lactobacillus grows out in the lupus-prone mice. So we looked at this Clostridialis lactobacillus balance and wondered if there's a dysbiotic state in human patients with lupus. And we have not yet done an interventional study with lupus patients, but we followed a small cohort of lupus patients at Yale. And we saw, interestingly, that healthy donors have sufficient amounts of Clostridialis, but in the lupus patients, we had to separate them into lactobacillus colonized patients versus those without lactobacilli. And interestingly, in this subset of patients with lactobacilli, we saw a loss of Clostridialis. So similar to essentially what we've seen in the mouse model, that the lupus-prone mice have a reduction in these short-chain fatty acid-producing bacteria, but with of course, as expected, a complicated situation in humans, that it's not all the same in one disease, but there are subsets of patients that have the same dysbiosis as in these mice. Now, obviously, we think that a personalized approach to diet in lupus patients would be useful with specifically targeting this subset that has this dysbiosis that may benefit from a resistant starch diet, which we have not yet given, but plan to do so in the future. So in summary, for this part, we think there's this actual dysbiosis, a term that's often used in the microbiome field, but ill-defined. We think in our case, with validated mechanistic links in the mouse model, there is this disbalance with a loss of Clostridialis and outgrowth of Lactobacillus reuteri. And we think that short-chain fatty acids can, through the resistant starch diet, can correct this imbalance. The question is what would be the mechanisms? And short-chain fatty acids and its effects on the host have been nicely studied by many in the field, such as Charles Mackay, who wrote this review several years back in Immunity, who studied this in asthma and type 1 diabetes, for instance. And it's well known that, for instance, also Wendy Garrett has shown this in colitis, that short-chain fatty acids promote regulatory T cells in the gut, and they can be anti-inflammatory. It's also known that the epithelial integrity is increased by short-chain fatty acids. And so we asked if these two mechanisms are at play in the lupus model fed with this starch. Interestingly, we saw no effect on T regulatory T cell levels. So there's probably resistance genetically or otherwise in these lupus-prone mice, as we know also in patients. The short-chain fatty acids had no effect on this adaptive immune cell arm, but it did improve epithelial integrity. And this was important because we studied also the barrier function in these TLR7 overexpressing mice and saw that TLR7 actually impairs the barrier function or other signals that mediate TLR7 activation. And that's shown here in a more complicated data slide I want to walk you through, that resistant starch improves TLR7 mediated gut leakiness. What you see here is that we've studied germ-free mice treated with a topical TLR7 agonist called imiquimod, and they're either just gavaged with a culture medium for Lactobacillus reuteri, MRS, or with the actual pathobiron Lactobacillus reuteri, LR. And you see with fitsidextran measured in the blood after we gavaged fitsidextran in the gut and measured a few hours later to see if it leaked through, that TLR7 activation itself with imiquimod greatly increases barrier leakiness, but that gavage with a pathobion that translocates further worsened gut barrier leakiness. And importantly, that resistant starch improves both of these mechanisms, the host inflammation and gut microbiota mediated gut barrier leakiness. In both cases, resistant starch suppressed gut barrier leakiness. So it did contribute to gut epithelial integrity. Furthermore, we tested if perhaps short-chain fatty acids could have a direct effect on the pathobiome, in our case, Lactobacillus reuteri. And in vitro, remarkably, indeed, Daniel could show that butyrate directly had growth inhibitory effect on Lactobacillus reuteri, which I think is still a little explored in the short-chain fatty acid field. There's, I think, some data on Candida suppression, but generally we don't know well how short-chain fatty acids may directly act on the microbiota. There was, by the way, a nice study from Wisconsin around the same time published showing that actually short-chain fatty acids may induce phages in L. reuteri and lactobacilli, and perhaps that's one of the mechanisms how growth inhibition occurs. In either case, we think that gut barrier integrity as well as direct growth inhibition are two mechanisms how resistant starch induced short-chain fatty acids improve the pathogenesis of lupus. And this fit, with the in vivo data, going back to the TLR7 transgenic mouse model, where we know these PDCs, these bad immune cells, are enriched, and short-chain fatty acids directly fed to these mice had significantly suppressed this PDC accumulation. So in summary, Daniel Zegarra-Ruiz showed in his PhD thesis that resistant starch type 2 like diet in mice will be fermented in this lupus-prone mouse model by many bacteria that are known to produce short-chain fatty acids, among them Clostridiales. This happens probably not only in the large intestine, but also in the small intestine, where we have an outgrowth of Lactobacillus reuteri that translocates and promotes the type 1 interferon signature. But the short-chain fatty acids have a direct growth inhibitory effect. And importantly, probably more important, it has a gut barrier sealing effect, because otherwise in this inflammatory TLR7 overexpressing environment, these pathobiomes translocate to extraintestinal tissues such as the spleen, and they can activate or hyperactivate PDCs, these innate cells that spew out type 1 interferon, which is a key signature cytokine in lupus. All right, so we still wonder, of course, a lot, how can we translate this to humans? And fiber studies in healthy humans have been done nicely by others in the field. For instance, here's a nice study where undergraduates were tested for their microbiome under a fiber diet. And you can see, which fits well with the literature, that bifidobacteria are often growing out in healthy subjects. And we saw also in the lupus-prone mice, besides the Clostridiales, that among the high enriched bacteria with the resistant starch diet were bifidobacteria. So we asked if perhaps in our lupus cohort at Yale, if we could find similar changes of improved gut microbiota communities. And with we, I mean here Irina Kulyuk, physician at Indiana University, who works with us on this question, as well as Marcia Pereira, a postdoc in the lab at the University of Münster, where we currently conduct this research. And Irina took the dietary history, which we obtained in this lupus cohort, and looked at the amount of resistant starch in these patients. And we not only looked at the systemic lupus patients, but also at a lupus-related disease called antiphospholipid syndrome, or APS, that we studied cross-reactivity previously in the paper I mentioned at the very beginning. And this is not an interventional study. This is, it's quite coarse. We were just looking at what did the patients eat. But Irina could nicely separate their diets into low and medium resistant starch intake. There was no patient that had high resistant starch intake, but there was a nice distribution, even distribution of low to medium in these patients with lupus and antiphospholipid syndrome. And what Marcia then saw when she looked at the microbiome community structure is that there was a direct and strong correlation between the resistant starch content and bifidobacteria. So this is the first hint that fits with this other human study in healthy undergraduates, and partly with some of our mouse microbiome data, where we also saw bifido outgrowth with a starch diet. Of course, the actual dysbiotic improvement of suppression of lactobacilli, outgrowth of short-chain fatty acid producing bacteria, we were curious about, but we think we need an actual interventional study with higher doses of resistant starch than what's in the natural diets of these patients that we track. So our goal is to eventually do an interventional resistant starch study in lupus patients. And what's very nice is that the field progressed again with healthy subjects nicely by a wonderful study by Justin Sonnenberg, who also speaks at this summit and may have talked about his study that came out last year. I just want to focus on one particular interesting aspect for us. So he published last year this healthy, mostly female subjects in the Stanford area in California, where they did all kind of omics besides the microbiome and food logs. They ramped up the fiber content. They didn't study specifically resistant starch, but fiber content, which went up from 20 to about 40 grams per day. And then after the maintenance phase, it dropped again. And why I'm really excited about this study is that it fits with what we've seen in autoimmune settings, that of course, there's a personalized response to the diets and also a personalized immune response. So what they saw is that the high fiber diet increased carbohydrate active enzymes and short-chain fatty acids, but that the immune response is personalized, that there were 3 subgroups of inflammation, anti-inflammation changes that correlated with the diversity of the microbiota. And most importantly, they did also immune signaling and tried to correlate this with these Cas enzymes, these carbohydrate active enzymes. And they looked at phosphostat signaling. And when you look at STAT1 signaling that's activated after interferon alpha, they saw a significant downregulation of this type 1 interferon signature, phosphostat1 signaling, with the fiber diet. So we think this is a nice suggestion that perhaps we are on the right track when we go to patients that we want to feed resistant starch, that perhaps in a subset of the patients, at least, we're hoping to see a similar beneficial effect on the interferon signature. So that's the outlook. As mentioned, we are now trying to develop with industry colleagues a resistant starch snack, because again, green bananas and raw potatoes is not something patients can easily eat. And with this snack, we hope to see a similar improvement of symbions in the microbiome of lupus patients that then may suppress pathobionts such as lactobacilli and enterococci, and thereby may prevent gut barrier translocation and promotion of the type 1 interferon signature. All right, so we think this is, of course, just the tip of the iceberg. I showed you some nice studies with diet in healthy human subjects. Dietary interventions are probably going to be in the future very interesting in a variety of chronic diseases. But I think the lesson is that we need to approach this in an almost personalized way, or at least that we need to look at subsets of patients within a given disease. And that may also be one of the reasons why many diet trials failed in the past, because you try to lump them all together in one disease, while their microbiomes and also host immune signatures may be subsets that respond differently to each diet. All right, that's my last slide. I'd like to just summarize that, of course, we are colonized with a microbiota at every barrier organ, not just the gut, but also the skin and the lung. And these microbiota, as mentioned in the beginning, are of course influenced by the environment heavily and by our diet. And if there's a dysbiosis, a disbalance between these pathobionts and symbionts, this can lead to direct barrier damage, but it could be just transient inflammation if you don't have the wrong genetic predisposition. However, the paradigm example for IBD, of course, is if you have the wrong genes affecting the barrier integrity, such as NOD2 polymorphisms that lead to an overshooting adaptive immune response to the microbiota, you can be predisposed to chronic inflammation in the gut and IBD. IL-23 receptor polymorphisms are well described in psoriasis, another important barrier immune factor that may contribute to dysbiosis and chronic inflammation in this skin disease. We hypothesize in this review here. And more speculative than mucin gene polymorphisms exist in interstitial lung disease, and there's clearly a deranged lung barrier in chronic inflammatory diseases that also may associate with a dysbiotic microbiota. But what I hope I've shown you today, that beyond these barrier organ diseases, outside the gut, there are diseases such as lupus, non-gut autoimmune diseases that may also be influenced by the microbiota if the barrier is leaky and if bacteria translocate and promote pathways such as the host pathway I introduced you to, the PDC type 1 interferon pathway. And specifically in the lupus mouse model, I have shown you that there's this disbalance between lactobacilli and the Clostridialis and other short-chain fatty acid producing bacteria, but with the right diet, at least experimentally, we could prevent disease by enriching again these good bacteria that produce short-chain fatty acids and seal the gut barrier, and even have a direct growth inhibitory effect on some pathobionts. With that, I want to close and want to thank again the people that did the work I've shown you today. I essentially mentioned them as I went along. Most of the work was done still at Yale, and Daniel Segarra-Ruiz, as well as Asma Al-Baidak and Rebecca Fine, were the main contributors to the diet study in mice. And the unpublished preliminary work we've done on the dietary history of the lupus patients at Yale was done at the University of Münster by Marcia Pereira and Irina Kuhl. And with that, I thank you for your attention. And if there's not a chat after the talk in the virtual summit, you should feel free to email me either at my Münster or Yale email accounts. Thank you.